My name is Shazni Damani, and I'm currently an undergrad student at the University of North Texas working with Dr. Wang. So as you saw previously, today's topic is protein purification. And a lot of y'all might be wondering what it is. What is protein purification? And the definition, kind of in layman terms, is that it's a series of processes intended to isolate one or a few proteins from a complex mixture. So what we're going to do today is actually show you step by step of how we do that in our lab. My lovely lab instructor, she got her whole gloves on, her whole lab coat on. And since, she wears glasses, she don't need no goggles, but, wear goggles. Hey guys, so today we're making luria broth, it's LB broth that we're making for our proteins. So we're just gonna measure out some water. Milky water, which is actually deionized water. Pretty much the same thing, but it's a little better. It's more pure. So we're going to show you how to read a graduated cylinder as well. We're going to see that the line, if you see, it's like just below 90. So now we're going to take it to our flask that has our broth powder in it. We're just going to put water in here. Make sure you see, it's not touching the sides, because we don't want the water to be anywhere but the actual agar is. So we're gonna, swirl it around. So now we are just putting some aluminum foil on it, so when we autoclave it, it doesn't explode, pretty much. We're gonna put some tape on it that will let us know that it's been autoclaved correctly. And as y'all have seen before on my snap, once it comes out truly autoclaved, the colors will be different as well for the big ones and the small ones. Taping it, the autoclave tape y'all have seen before. You see the little lines. I don't know if you can. I think you can see it with this light. The two lines will be black when we actually autoclave it, so it'll let us know it's been done perfectly and there hasn't been any harm to it. And now we take it and put it on our cart. Then we need to go upstairs and autoclave it, put it in so our samples will be done correctly. Back again at it here. Put our stuff. Autoclave. Is anything in there? So first things first, what we're going to do is we're going to check if actually anything's in there. It says operating mode. Usually that means somebody left anything in there. So what we're going to do, we're going to put on our handy dandy gloves so we don't get burned. We're going to open it, and yeah, there's nothing in here. I guess it's just being weird. We're going to put our samples in here. So we're going to get it all the way. Our samples and there. Our samples are in here. We're gonna close it up. Make sure it locks. We're gonna do liquid 20. We see that we're gonna put it on for 4, and we're gonna start the cycle. And the autoclave process has started. So we'll see y'all back here in an hour. We have our luria broth all ready to go. Look at that with our little cells. But that's the centrifuge we use. You see how big this is? Bam. This is where we're gonna put our samples that have been labeled. It is heavy. Dude, I have a strength of a chicken nugget. Oh, yeah, yup. That was heavy. So that's when we started it. We're gonna wait until it gets up to 4,000 for about 20 minutes. So we're gonna wait until you see the RPM. until it gets up to 4,000, and then we make our journey back. Y'all, so now we back again. Now it looks reddish. See, that's the pellet. That's what we actually need. And then we didn't need any of the broth. All right.
That's where our protein is. We don't want the supernatant, so we're just gonna discard that.
So the supernatant is actually the broth. So the liquid part of it that she's pouring back into the beaker. And she's pouring it a certain way because
you want to keep it away from like the flow of just this liquid, so it doesn't, fall out or something. And I usually leave the pellet facing upward, so like you're not accidentally
So that's our little pellets. You see they're red. And why are they red?
To his protein attaches to an iron.
And iron, as y'all know, when you see stuff rust, it is yellow or orange. And the reason why it's orange is because oxygen. Watch her pour. What we doing?
This is just licensed buffer. 95 microliters of substrate. It's not that much. We just want 0.1 millimolar concentration. And then mix it a little bit. So that we can start going. So you want to keep this on ice. What I'll usually do is pour a little bit of the buffer in there already. This kind of scoop. We're gonna just take out the pellet and pour it into there. If we want to like freeze it for later, we'll store it into the -80, which is what I'm gonna do with the other one. Just kind of get that out of there. Try to get as much of the pellet as possible. So like there's some liquid in there, kind of like shake it to get some off of the rest of the wall.